Okay, so we will start with fowl pox. So fowl pox is a very important disease of birds, and in particularly chickens, they have two important form. First one is a cutaneous form, and second one is a diphtheric form. Cutaneous forms are generally occurred when skin is involved, and diphtheric form is when the particular internal organs like upper respiratory tract and upper GI tract when they are involved. Then this form is known as diphtheric form. So generally, sorry, in cutaneous form, what you will find discrete nodular proliferative lesions on non-feather part of the body, and in case of diphtheric form, you will find fibrino-necrotic as well as proliferative lesions in uh, upper respiratory tract, mouth, and esophagus. Now, etiology. So fowl pox virus is responsible for this fowl pox disease. So this fowl pox virus is a double standard DNA virus, belongs to genus Avipox virus, family Poxviridae. Generally, these pox virus are very large viruses, and many a time they can be visible under light microscope. And particularly for fowl pox viruses, you have to remember two important terms: Borel body and Bollinger body. Right. So here you can see this is Borel body. So what do you mean by Borel body? Borel body are nothing but they are mature virus, and this mature virus particles, individual mature virus particle is known as Borel body, or they are also known as elementary body. Right. And the second term is Bollinger bodies. So Bollinger bodies are nothing but this is actually viral inclusions then can be seen in cytoplasm of the infected birds, cytoplasm of the infected cells, particularly epidermal cells. And this particularly Bollinger bodies are nothing but they are actually aggregations of some defective viral replication protein, and sometimes it may also contain Borel bodies, right? So Borel body, individual virus particle; Bollinger body, intracytoplasmic inclusion body. Now host. So among almost 9,000 species of the birds, 232 species are showing natural infections of fowl pox virus. So you can imagine how large species of the birds are susceptible for infection of this particular disease. Here you can see some list of this birds, species of the birds which are susceptible for infection of fowl pox viruses, and among that this particular canary birds are showing some highest mortality. In particular such birds, mortality rate may go up to 80 to 100%, while particularly in other birds generally mortality rate is low and you will find high amount of morbidity. Now transmission. So depending upon the form of the disease, you will find different type of transmissions. But generally, first we'll discuss about cutaneous form. So we have already discussed in case of cutaneous form, generally you will find skin lesions. So generally, in case of cutaneous form, suppose you particularly if healthy birds have some lacerations or some skin lesions or some lesions over the skin at that time, particularly bird handlers which have handled these affected birds before, and when they handle such type of a lacerated or some skin lesions birds, in that case they are responsible for mechanical transfer of this virus from affected to healthy birds. So generally injured skins are prone to this particular disease, as well as some particularly biting insects like mosquitoes, ticks, and mites. They are also responsible for transmission of this disease, but this is done just simple mechanical transmissions, right? So there are two type of transmissions: mechanical transmissions and biological transmission that can be used for some vectors. So generally in case of mechanical transmissions, there will be no virus replication occurs inside the vector, and here the same thing will happen. Generally this biting insects like mosquitoes, ticks, and mites, they will try to take the feed on infected birds, and at that time this virus will get attached with this vectors, and then they will come into the healthy birds, and at that time they will transmit this particular disease into the healthy birds. So this is route of transmission in case of cutaneous form. And the second form is diphtheritic form. Here generally what happens, generally inhalation or ingestion of the virus is responsible for transmission of this disease. In this particular form you will find lesions in internal organs rather than skin. But in one study which is conducted very long back, particularly in India, and that show generally in any route of infection, generally virus will reach into the internal organs, they are replicated inside the internal organs, and then they will produce disease either in skin or in internal organs. But generally in any route, virus will replicate inside the internal organs like spleen, liver and other organs. Now pathogenesis. The pathogenesis which is mentioned in this particular slide is actually generalized. They are not directly related with the pox virus. But if we talk about any pox virus, not only avian pox or fowl pox viruses, but when we talk about any pox viruses, these pox viruses are responsible for two important features. They are responsible for necrosis of the epithelial cells, as well as they are also responsible for proliferation of the epidermal cells. Right? So here what happens, when the virus will replicate inside the epithelial cells or inside the infected cells, they will induce some degenerative changes, as well as they will induce necrotic changes. So you will find necrosis, as well as there are some important virus like molluscum. This particularly molluscum pox, this is again molluscum is a pox virus, right? So these molluscums have some important proteins which act like a epidermal growth factor. That particular protein will act as a epidermal growth factor, and we all know this epidermal growth factors are responsible for proliferation of epidermal cells. So this particularly viral protein will act as epidermal growth factor, and ultimately they are responsible for proliferation of epidermal cells. So in most of this pox virus infection, you will find both type of lesions, necrotic as well as proliferative. Right, and this is the reason why you will find such type of changes, because this particular virus, when they replicate inside the cells, they will induce necrosis, as well as particularly some virus contains some typical proteins which act as a epidermal growth factor, or you can say there are some their structures are somewhat similar with the epidermal growth factors, and they will induce mitotic activity of the epidermal cells, and ultimately you will find proliferation of the epidermal cells. Right, so this is the reason why you will find such type of lesions. Another important thing when we talk about pox viruses, the development of the lesion is very unique. Right, here generally what happen, either through any route, either through aerosol routes or through inhalation route or through infected skin. Initially this particular virus will enter into the body and it will infect macrophages and the lymphocytes, and through this and lymphocyte trafficking systems, ultimately it will reach into the different organs. Initially they will replicate, and then finally they will again come into the skin part, and initially in the skin it will replicate initially in the endothelial cells and will induce some abnormalities in the endothelial cells. So there will increase vascular permeability, so there will be diffusions of the fluid, as well as some RBC will be come out from the infected blood vessels. So initially you will find some maculae. Right, so macula is nothing but this is non-raised, slightly reddish color area which can be seen in skin, and then after this particular from this endothelial cells, this particular virus will infect Langerhans cells, and this Langerhans cells will reach into the epidermis and they will infect keratinocytes or other cells of the mucosa or epidermis, and this particular virus will replicate inside the such type of cells. If we talk about epidermis, then it will replicate in the stratum basale and stratum spinosums. So in stratum basale and stratum spinosums, initially this particular cells will replicate and it will induce some vesicular degenerations. After some time, such type of infected cells will get lysed, it will get necrosed, and ultimately you will find some small vesicles, and such type of small vesicles are known as reticular degeneration. And during these replications, this particular virus infected cells will release variety of cytokines, and ultimately this particularly cytokines will attract more and more number of neutrophils or heterophils in case of birds at the site of vesicular degenerations, or sorry, at the site of reticular degenerations. And generally slowly and slowly this particularly inflammatory cells will travel into the vesicles. So initially you will find papules, then vesicles, then when these neutrophils will enter into the vesicles, you will find pustules, and slowly these pustules will get ruptured, and this necrotic debris will come out over the surface, and you will find scabs over the such type of lesions. As well as we have already discussed, that's particularly some viral protein of this virus will act, or they have some homology with this epidermal growth factor, and ultimately it will induce hyperplasia of the epidermal cells. So you will also find hyperplasia surrounding the lesions. So generally what you will find, lesions in center will show necrotic changes, while periphery generally will show slightly upper slightly elevated margins due to hyperplasia, and probably sometime it will be red in color, and such type of lesions are known as pock lesions, pock lesions, right? So here also most of these pathogenesis are almost similar. Now clinical sign. So incubation period is ranging from 4 to 10 days, and they again depend upon variety of factors. And this particularly disease is they have two forms, cutaneous form or dry pox form, and diphtheric form or wet pox form. But generally these clinical signs are somewhat variable depending upon the host species, the virulency of the virus strain, distributions of the lesions, and other complicating factors. So in case of cutaneous form, when the skin is involved, most commonly this generally cutaneous form is most common whenever you will find outbreaks. Generally you will begin, these lesions are begin with the pimples or papules like lesions or scab like lesions on non-feathered area of the body, particularly like skin, skin of comb, wattle, eyelids and legs. And particularly in case of turkeys, this particularly such type of papule to scab like lesions are also extended into the feathered areas. And due to this particularly such type of lesions, generally birds will have some difficulty in their feeding. It will take a less food, as well as their weight gain will be also decreased, or occasionally it will also show weight loss. In diphtheric form, generally lesions are not much typical outside the body, but generally birds birds become lethargic. It will not taking proper amount of feed or water. Sometimes when this larynx or trachea are involved, at that case these particular lesions will prevent proper respiration, so you will find difficulty in breathing. You will also find nasals or ocular discharge. So majority of these gross pictures are taken from Cornell University website for education purpose only, and here you can see this is beautiful pictures that showing comb lesions, right? So this is hair, non-feathered part, and here you can see the skins are showing two type lesions, center area of necrosis and somewhat proliferation in the periphery, right? So this is typical pox lesions. Here you can see these pox lesions are involved comb, wattle, beak and periocular tissue, right? So here also you can see majority of these are very hard nodule like lesions, right? So this is nothing but due to this proliferation of epidermal cells. Here you can see this is another pictures where these combs and wattles and some this particular skin and periocular tissue are get affected. These are typical pox lesions, and they are showing this is little bit chronic lesions. And many a time what happen when these chronic lesions occur, many a time when they get healed, at that case generally the pigmentation will be lost. Here you can see this is a little bit more hemorrhagic lesions, more scabs are present over the lesions. But again, this is necrotic as well as proliferative lesions that involve comb petals and beak as well as periocular tissue, and this is majority are non-feather part of the body. Here also almost similar lesions. You will find some oculonasal discharge, ocular discharge. Right, so here involved combs and wattles. Here also almost similar lesions involve most of the non-feather part of the body. So nodular proliferative lesions as well as you will find some necrosis. Here you can see this is little bit chronic lesions where these nodules are become very large. These proliferations become uh, more severe. You can see this entire conjunctiva are converting into the proliferative nodular mass, and combs and wattles are also severely affected. Here you can see generally this non-feathered parts are all feathered parts are also involved, but in most of the cases generally turkey will show such type of lesions. Now gross lesions. So in cutaneous pox or dry pox, generally you will find variety of depending on the stage of disease, you will find some different type of lesions, but most commonly you will find papular lesions on unfeathered skin of the head, neck and feet. That may also involve combs, wattles, beak, periocular tissues as well as the toes and legs are also involved. Feet are also involved, and general lesions can progress from papules. So you can find the papules, vesicles, pustules, and when these pustules are get ruptured, you will find scab over the lesions, and slowly and slowly you will find proliferative lesions, right? So depending upon the stage of lesions, you will find different type of lesions, and we have already discussed particularly when such type of lesions are get healed, generally at that particular area pigments will be lost, and generally you will find discolored, you will find some discoloration at that particular site. In diphtheric form, generally skin are not much involved. Generally, majority of the lesions are seen in internal organs, and you will find small white nodules in the tongue, esophagus, pharynx, and larynx. And these nodules sometimes get coalesced, they get mixed with each other due to when they get enlarged. Two near nodules become one single large nodules, and this particularly may show yellowish or whitish colors, and it may contain some fibrin material, right? So you will find some fibrino-necrotic and proliferative lesions in mucous membrane, right? So this is cutaneous form of pox, and we have already discussed in clinical science. You will find such type of proliferative as well as necrotic lesions. And here you can see this particular image. The feathers are actually removed for smooth appreciation of the lesions, right? So here you can see you will find some large nodules, and they are again nothing but they are actually large proliferative nodules, which is due to viral protein effect. Here you can see these feet are involved, particularly interdigital space are also involved, where you will find such type of proliferative lesions. And now this is weight form or diphtheric form, where internal organs are involved, and this is esophagus, right? So here esophagus, you will find such type of white nodules in esophagus. You can see this multifocal to coalescing white nodules that involve esophagus. In this case, you can see this is trachea, and trachea is filled with the fibrino-necrotic material, and in this case you should have one differential diagnosis from the ILT, infectious laryngotracheitis, and if this is not if we are unable to confirm on basis of gross lesion, in that case you have to go for macroscopic examination, particularly in case of ILT. This particularly sloughed epithelial cells which show some intranuclear inclusion body, while in this case generally you will find cytoplasmic inclusion, particularly in cells which showing viral replication. Here also you can see this trachea is filled with the So necrotic and fibrinous exudate. Okay, then just like ILT, this pox virus are also responsible for production of POC lesions, POCK POC lesions, right? So generally here what happen, and these particular inoculation are generally done for virus isolation. So if you want to go for virus isolation, in that case, what you have to do, you have to inoculate this particular virus into the embryonated chickens, and after a few days you will find such type of proliferative lesions. And with what we have already discussed, generally such type of lesions have a center area which is get depressed due to necrosis, and surrounding lesions will show proliferative lesions. So such type of lesions are known as POC lesions, and such type of lesions are generally found in chorioallantoic membrane of the embryonated chicken's egg. Right. So for this part, if you want to go for virus isolation, in that case, what you have to do? You have to isolate this particular virus, and then approximately 0.1 ml of tissue suspension of skin or diphtheritic lesions with appropriate concentration of antibiotic is inoculated into the chorioallantoic membrane of a 9 to 12 day old developing chicken's embryo. These are incubated at 37 degrees for 5 to 7 days, and then examine for focal white POC lesions. What we have already discussed here in this picture, they are visible, right? Here you can see these are the POC lesions, right? Histopathologically, what you will find? You will find eosinophilic intracytoplasmic inclusion body. In this particular area, the center area will show necrosis and periphery will show slight hyperplasia. Here you can see this is in this particularly skin are showing hyperplastic lesions, right? This is skin of embryo, they are showing hyperplastic lesions, right? Now microscopic picture. So microscopically, what you will find variety of lesions starting from ballooning degeneration to reticular degeneration, papules, then vesicles, pustules, square formation, right, as well as epithelial hyperplasia. The most important thing that you will find in case of fowl pox virus infection is an intracytoplasmic eosinophilic inclusion body, right? Because this virus are replicated inside the cytoplasm, so you will find intracytoplasmic eosinophilic inclusion body, and this inclusion body are known as Bollinger bodies, right? We have already discussed there are two words, Borel bodies and Bollinger bodies. Borel bodies are nothing but they are actually mature individual virus particles, and the Bollinger bodies, they are actually intracytoplasmic eosinophilic inclusion body. And whenever you find such type of very prominent inclusion bodies, they are typically indicative of fowl pox. Here you can see this particular macroscopic picture is taken from Cell Davis Foundation page, and here you can see this is one cell. You can see, you can easily appreciate the cell border, and this is nucleus, this is nucleoli, and you will find eosinophilic intracytoplasmic eosinophilic inclusion body. So this is Bollinger body, and whenever you find such type of Bollinger body, this is very characteristic features of this fowl pox. Majority of the cells are showing such type of inclusion. So now diagnosis. Generally, gross lesions are majority of time whenever you find cutaneous lesions. Cutaneous lesions are very characteristic, and generally they are indicative of this pox lesions. But in case of cutaneous lesions, generally you have to remember, you have to go for some differential diagnosis. Like in case of trachea, when you will find you only tracheal lesions, in that case you have to go for differential diagnosis with the ILT. And particularly in case of doves and pigeons, particularly when another infections like trichomonas infection, cankers, you will also go for differential diagnosis. Generally, if you go for microscopic examination, then you can easily differentiate between these two entity. Then one another methods, particularly Gimenez stain, can be used for impression smear. In that elementary bodies, particularly individual mature viruses, can be stained with the Gimenez stain. So you can again you can go for differential diagnosis with the help of Gimenez stain. Electron microscopy, as well as if you go for virus isolations, in that case you will find pox lesions in chorioallantoic membrane, as well as for serological tests like virus neutralization test, agargel immunodiffusion test, ELISA, PCR immunohistochemistry can be done for confirmation of this disease. But most of the time, gross lesions are very much characteristic, and rarely you have to go for microscopic examination. So this is all about pox virus. Thank you.